These are the lecture notes for the chapter on viruses. This is chapter 1 for Bio 112. So viruses are not actually considered living things. We do study viruses in Bio 112 because obviously viruses affect living things and they have similarities to living things. But biologists do not consider them to actually be a living organism. That's why they're not classified, into a domain or a kingdom or a phylum or anything like that. So we are going to learn about viruses. We have a whole chapter, but and we're going to learn how viruses affect living things and that kind of thing. That they are not considered an organism. They're not considered a living thing. Okay, so what we know about viruses is they are acellular and they are parasites. It says they're parasitic entities. That means they're parasites. They're not classified within a kingdom or a domain, actually. They're not cells and they can't divide. So what they do is they reproduce by infecting a host cell and kind of tricking that host cell into making copies of the virus. So viruses can infect all types of living organisms. Viruses can infect bacteria, protozoans, algae, fungi, plants, and animals. In fact, the first virus that we're going to talk about is the tobacco mosaic virus, TMV, tobacco mosaic virus, and this is an electron microscope image of the tobacco mosaic virus. This is a plant virus that infects lots of different plants, not just the tobacco. In fact, you can see here in the second picture, the picture on the left, these are images of orchid leaves. If you are from this area, you know that's not tobacco. But these are orchid leaves, and it shows kind of what they look like when they're infected with the tobacco mosaic virus. I know that it can also infect tomato plants and, of course, tobacco, but it can infect different types of plants. Okay, so are viruses alive? We've already said no. It says viruses exist in a netherworld between a living organism and a non-living entity. So, in other words, it has characteristics in common with living things, but doesn't have all of the characteristics that something must have to be considered an organism. So it says living things grow, metabolize, and reproduce. Viruses replicate, but they are dependent on their host cells to replicate. And they do not metabolize or grow, but they're assembled in their mature form. So they don't have any stages, they don't go from, like humans go from being a baby to being an adult and go through the different life stages, and plants go from being a seed, and to growing into a mature plant. Well, viruses are just, they just have one stage. Viruses basically consist of two main structures, two main parts. And we learned this in Bio 111. I don't know if you remember this or not, but they have a nucleic acid core, and their nucleic acid core can either be DNA or RNA. So they have either DNA or RNA inside their core. And this is interesting because we learned in Bio 111 that DNA is double-stranded and RNA is single-stranded. But in viruses, the DNA can be single or double-stranded. And we write it like this. We'll go, if it's single-stranded DNA, we'll write it ssDNA. And if it's double-stranded DNA, we'll write it like this. And RNA can be the same exact way. It can be single-stranded RNA or double-stranded RNA, whereas in all other organisms, the DNA is the only molecule that's double-stranded, and RNA is always single-stranded. So we know that viruses consist of genetic material, either DNA or RNA, but they also consist of a capsid, and the capsid is composed of protein. So viruses consist of DNA and they consist of protein, and sometimes they'll have an envelope on the outside. Now, Viruses were not discovered until the 1930s because you cannot see a virus without using an electron microscope. This image, picture A, is actually an image of a portion. This little triangle portion is just a part of an E. coli bacterium. So this is definitely an image taken with an electron microscope. And then the virus is here, and the virus kind of has a head, and then it looks like little legs that help. It's called the tail portion. The tail portion has little appendages, or they look like legs. It kind of, to me, looks like a spaceship. But anyway, the DNA is in this head region. And then the legs actually help it attach. The tail portion actually helps it attach to the E. coli bacterium. So just to show you about size, this is a light microscope image. Picture B is taken with a light microscope like we use in lab. And this is the inside of the intestines. This is just one single intestinal cell from a mammal. It's probably a human. You can even see it looks like the nucleus right here. So this is one human cell. This is an individual E. coli bacterium. We learned that they were rod-shaped. So this shows you the E. coli bacterium, how big it is compared to a eukaryotic cell. So you definitely couldn't see a virus on the surface of an E. coli bacterium with a light microscope. Okay, now, I don't like this word theories because they are not theories, they are hypotheses, and there's a difference. We learned that in Bio 111, so I'm crossing that out to begin with, but we do have 3 different hypotheses on how viruses originated, the evolution of viruses. One hypothesis is called the devolution. I think that's how you pronounce it, the devolution hypothesis. I know it says regressive also, but I've looked through your quiz questions, and your quizzes use the term devolution. So the devolution hypothesis is related to evolution. It proposes that viruses evolved from free living cells. Which makes sense because viruses contain, genetic material, nucleic acid, and they also contain proteins. And sometimes they even contain an envelope that's very similar to a plasma membrane. It's got like a lipid bilayer and proteins within the lipid bilayer. So that's the devolution hypothesis. The escapist hypothesis accounts for viruses that have either an RNA or DNA genome and suggests that viruses originated from RNA and DNA molecules that escaped from a host cell. That's where the escaped comes from. So this hypothesis is really more related to the genome or the genetic content of the virus. And which in that case, the genetic content would have the code for any of the other parts of the virus, like the protein capsid and any enzymes that are needed. Then there's the self-replication hypothesis, which hypothesizes that these viruses self-replicated, maybe alongside maybe a process like coevolution, kind of like similar to the coevolution of flowers and insect pollinators, but this hypothesis just suggests that they self-replicate, but this occurred alongside their host cells. So they're self-replicating molecules likely evolving alongside the cells that they rely on as hosts. Okay, viral morphology. The word morphology just means structure. We know that a virus does not have a cellular structure. When we are speaking of a single virus, we use the term virion. A virion consists of a nucleic acid core. We've already discussed that could be RNA or DNA, single-stranded or double-stranded, and then an outer protein capsid. Sometimes viruses only contain a capsid, and we call those naked. And then other viruses have an envelope outside the capsid. And if they have an envelope, the envelope is actually composed of a phospholipid bilayer and protein embedded within the phospholipid bilayer. And it's so similar to the host cell that we believe that the envelope was derived actually from the host cell that the virus infects. Sometimes viruses will contain additional proteins, such as enzymes, like DNA polymerase, RNA polymerase, some of the enzymes involved in protein synthesis. The core of the virus contains the genetic content of the virus. The genetic content, we already said, can be either single or double-stranded, but let's see. But also the chromosomes that form can either be linear or circular. If you remember, bacterial chromosomes are circular. Human chromosomes are linear. So that's another way we can classify viruses. The capsid is composed of capsomeres, and that's important. That's in one of your, I don't know if it's a quiz question or a practice question, but it does, you do have to know that capsomeres are the protein subunits that compose the capsid. All right, so comparing DNA versus RNA viruses. In DNA viruses, the viral DNA directs the host cell's replication proteins to synthesize new copies of the viral genome and to transcribe and translate that genome into viral proteins. So what this is saying is that if a virus is a DNA virus, it goes through the same process of transcription and translation that we learned in Bio 111. It contains DNA. It uses the host cell and the host cell's RNA polymerase to transcribe messenger RNA, and then that messenger RNA is translated into a protein. We learned that this is how protein synthesis occurs, but RNA viruses are different. So RNA viruses contain RNA. And what they have to do is that RNA either skips transcription, okay? So that RNA serves as the messenger RNA. Or in an even more complicated process, retroviruses have RNA that are converted into DNA. Then the DNA undergoes transcription, is converted to messenger RNA, and then the messenger RNA is translated into a protein. So it's like a step, and they have to use an enzyme called reverse transcriptase, because RNA being converted to DNA is the reverse of transcription. Let's see, just to make sure we've got all this material. Okay, RNA viruses contain RNA as their genetic material to replicate their genomes in the host cell. The RNA viruses encode enzymes that can replicate RNA into DNA. So, because that can't be done by the host cell. So they do have to have the genetic code for the enzymes to convert RNA into DNA. These RNA polymerase enzymes are more likely to make copying errors, so therefore there are more mutations occurring, more like spontaneous mutations occurring in RNA viruses. So this causes it to be very difficult to make a vaccine for these viruses. The flu virus, the cold virus, HIV, these are all viruses that have RNA, and it's very difficult to make a vaccine for these viruses. I think I'll stop here as we only have 30 seconds.